Hello, everyone. My name is Sharisa Martin. I am the Program Manager of Professional Development for the American Society for Cell Biology. I welcome you today to join us for our webinar, Communicating Science Through Art, in collaboration with the Public Information Committee. Here to share more about that committee and give our speaker introduction is Janet. Janet, please turn on your camera.
So my name is Janet Owasa. I'm an assistant professor at the University of Utah and a member of the Public Information Committee of the ASCB. So on behalf of the PIC, I'd like to welcome you to the second webinar of our series that we're calling Communicating Visually. So just a couple of things that if you're interested in general about visual communication, we're having, and you're attending the annual meeting in a couple of weeks, we're having a roundtable discussion on Monday, December 5th at 1 p.m. So we're going to be gathering together to talk about science visualization and research, communication and outreach. So please do stop by if you're at the meeting. And also, Beata will be mentioning this at the end. There is a subgroup session on Saturday that Beata has co-organized with a couple of other people. That's called Science and Art, Bridging Two Creative Universes. So that'll be held on Saturday at 1 p.m. So I'd like to introduce our speaker, Beata Mierzwa, who is a postdoctoral researcher at the Ludwig Institute for Cancer Research at the University of California, San Diego, where her focus has been to advance our understanding of cell division and improve cancer therapy. So beyond her academic career, Beata creates science-themed drawings and clothes. So you can see an example of that here. Her hand-drawn illustrations use metaphor to portray scientific concepts in intuitive ways, while her microscopy fashion celebrates the beauty of the molecular world. She's also created a science video game called Microscopia that invites players to explore the beauty inside our cells. And through her AAA ifthen ambassadorship for science outreach and her work with young women in biology in Southern California, she aims to inspire female students to pursue careers in STEM. So yeah, welcome Beata, and I'm looking forward to your presentation.
Thank you so much for the introduction. And thank you everyone for being here. I hope you can all see me and hear me. Huge thank you to everyone who's joining. And I'm super excited to chat with you all about the topic that I'm really passionate about, which is using art as a tool to communicate science. So my name is Beata. I'm a postdoc working on cell division at the Ludwig Institute for Cancer Research at the UCSD in San Diego. And in the lab, as Janet already mentioned, I study how human cells divide. And the aim is to improve the way we treat cancer. And along my academic career path, I also love creating science art and science fashion for science communication. And I'm also really passionate about science outreach. And I love using art and video games and student workshops to share the beauty of biology with the world and also get creative students excited about science. However, up until my PhD, I had no idea at all that combining science and art could be such a fantastic tool for science communication. So I'd love to tell you a little bit about how I discovered that I can combine my two passions, which was during my PhD, how I go about creating scientific illustrations and how I go about coming up with ideas, and also hopefully inspire you to experiment with some creative adventures as well. So science and art. Many of us probably grew up hearing that they have very little in common and then either you can be a scientist or an artist. But historically speaking, art and science were never completely separate. Drawings were really crucial to record and to communicate scientific findings, and therefore scientists really had to be artists as well. So, for example, Leonardo da Vinci, he brilliantly combined art and science and engineering, and the skills that he developed as an artist really enabled him to draw all of these super detailed representations about biology and anatomy and his inventions. Another really great example is Ernst Haeckel, who illustrated Darwin's ideas on evolution and on natural selection. And these drawings were crucial to make his work known to the public at that time, and was a completely new concept. And finally, here are some really gorgeous drawings of cells created by Walter Fleming. And he basically drew what he saw under the microscopes. And there were no cameras, there were no computers. So these drawings were really important to share his findings. And some of them are really amazingly accurate even today. So all of these people really were both scientists and artists. And their work really made a huge impact through their science art and through their communication. But nowadays, what are the benefits of doing science art? So in my own experience, discovering the combination of science art has had two big impacts. One of them is how I think about and communicate my own research. So for example, making illustrations has allowed me to practice a skill that I think is really important in science in general, which is, taking a complex manuscript or a complex set of data and portray the essence of the story in a clear and concise way so that everybody can make sense of it. Also, illustrating other people's research from different fields has been super helpful to gain new ideas for my own research. So often the starting point is a manuscript. And the first step in my creative process is to read the paper, read some background literature, and get familiar with the field. And this has led me to discover some really cool literature that I would have otherwise never read and got some ideas for my own research. And finally, promoting your science in artistic ways can help you get funding for your research and also encourage creative scientists to join your lab, because there's a lot of creativity required for science as well. And the other big impact was how I interact with people outside of the scientific community. So communicating science can really increase the impact of our discoveries. And we can show the world that what we do as scientists is not only fascinating, but also really beneficial to our world, especially now where these days this has become really important. And it's also a great way to give non-scientists a chance to appreciate the beauty and the wonder behind biology that they might have never seen before. And most importantly, it also has the potential to spark fascination and curiosity and really inspire our next generation of scientists, which is really important as well for our future. But when I started creating science art, I had none of this in mind. And I would have never thought that discovering my passion for art could turn out to be so helpful in so many ways in my research and beyond. So I want to go a little bit into detail about both of these aspects as I tell you how I found my way into making science art during my PhD. So let's start with the communicating research inside the scientific community. So when I was younger, I always loved art. And I was also kind of fascinated by the fact, how we are made of cells and how DNA is our genetic material. But at that time, it never even crossed my mind that I could combine science art, basically science and art. And because, I didn't have any role models who were doing this at this time, it's not something that was on my horizon at all. And so when I finished high school, I wasn't sure what to study. I loved art, I loved science. And I thought I had to choose between my two passions. So I decided to study molecular biology first. And I thought I could always continue doing some art at home, on the side, and things like that. But when I started studying, I fully focused on working in the lab and never had a chance to make time for creative projects. So I always thought, maybe one day I have time, but there was always something happening. There was always something that was more urgent. But then a few years into my PhD, I had a chance to create my first science drawing. And so we were making this big poster as a gift for the organizers of my PhD fellowship at that time. And each of us had a little square to fill with something that's related to our research project. And I thought this could be a really great opportunity to try something artistic and see how that goes. And at that time, I was working on a process called cytokinetic abscission, which is the final step in cell division where the last connection between the cells is cut. And this is what I drew. So you have two cells. They are still connected by a bridge, and they are literally using scissors to cut each other apart. So I thought it was kind of like a fun rendering of the process that I was studying. And I thought it'd be fun to try to use it to introduce my research questions in the title slide of my presentations. And I thought I would just try it out. And I actually specifically remember presenting it for the first time at the ASCB meeting back in 2015. And I realized something amazing. I noticed that people from conferences and from my institute, they would remember my research even years after I presented it. And that's because they remember the drawing, that there was something really unconventional about it, and together with it, the science that I was working on. And this is really the first time that I realized that science art could be an amazing tool to communicate my research. And this inspired me to continue experimenting. So I started playing with more different creative ways to present my research and my findings as they were emerging during my PhD. So this is the same drawing here, but this time it's overlaid with real microscopy images. So it highlights the DNA inside the nuclei and the microtubules that span the bridge that connects the cells. But of course, instead of using actual scissors, cells use a protein machinery that is called escort 3 to cut the bridge between them. And you can see in this movie what this process looks like. So you see how escort, which is labeled in green, first accumulates and then just right before the cut. And this happens twice, and it results in 2 separate cells and the remnant of the central part of this mid-body. But how does this actually work on a molecular level? So in more mechanistic terms, the idea is that during these stages, escort forms these spiral filaments that constricts the membrane until the cells can separate. So what you see here is what you saw before, but this time the bridge is shown vertically. So imagine there would be one cell on the top and the other one on the bottom. And I was thinking a lot about how to portray this concept. And at one point I had the chance to participate in a small art competition at our institute. And I thought it would be fun to maybe try to create a dress. And so I made this dress that's supposed to show exactly the same process. So you have the same color code, and you can see the green roams that are representing the filaments. And I'm actually wearing a corset to illustrate the concept of constriction, which narrows down the bridge until the membranes can separate. So I continued this experiment, and I wore it at the Vienna Science Ball, which is one of these fancy big events in Austria. And this one brings together scientists and people who are interested in science. And I couldn't believe how many people asked me about the meaning of this dress and how easy it was to get people curious and excited about my research just by presenting my science in such an unconventional way. And the actual topic of my PhD project was about investigating the mechanism of this constriction. And the way that this escort works is that each filament is not continuous, but it's built of many small subunits that assemble when they are needed during cell division. And when I started working on this, people generally thought that these subunits first assemble into stable filaments that then change their shape during constriction to narrow the membrane. And different mechanisms were proposed for that, and it was kind of the way people were thinking about it. But when I investigated the dynamics, we found something really surprising. We found that this structure is actually highly dynamic. So this is the only experiment I will show. But basically what you see here is a photobleaching experiment where I bleached the escort pool at the bridge and found that the fluorescence came back really rapidly, meaning that the subunits that form these polymers are constantly being exchanged inside the structure. And all of this turned out to depend on an enzyme called VPS4. And so this kind of was the key finding in my thesis, and this ended up being the model figure in my paper. So basically what it means, we have a structure that is made of all of these small building blocks, and it's being constantly remodeled. And so basically we have a structure that is strong enough to deform membranes, but at the same time also very dynamic, which is kind of counterintuitive and kind of hard to explain. But I thought instead of kind of using this model figure, I thought that everybody understands the simple concept of building blocks. So I started playing with Lego pieces, and I made this analogy to think of the subunits as building blocks. And these building blocks can be moved around by the Lego people, which represent the enzyme that is needed to keep these structures dynamic. And here's the final drawing to go along with the model figure from my paper. And it's basically supposed to be like an intuitive way of rendering the same concept. And so this is just an example for my PhD. And as I was experimenting with all of these experiences really made me realize that even if something is getting more and more complex, like, it always happens in science, there is always a way to make things more intuitive by using metaphors and connecting it with something that we are all familiar with from everyday life. And this inspired me to continue experimenting and to continue making drawings. And so while I was playing with all of these art projects, other people started asking me to make drawings for their research. First it was people from my lab, then other people from my institute who were working in different fields, and then also people who I didn't have a prior connection with. So I ended up drawing every night after coming home from the lab and on the weekends. And I made illustrations for papers and journal covers and conferences and even space missions. And it was just a lot of really fun and unexpected adventures. And I realized that all of these projects added a different perspective for me about how I think about science. And it also gave me a great balance in addition to the lab work that I was doing. So it's something that I didn't really expect when I started out. But the real surprise came when I started posting my artwork online. I was really amazed to find that I was not the only one doing this, and that there was a whole community around it. There were so many talented people creating all kinds of different art and having all kinds of different career paths. So for example, some scientists use art to promote their research with these renderings of the inside of the cell, with neurobiology and development. And some people are science illustrators, so they work with authors and journalists to create these gorgeous artwork for journal covers. Some people specialize in 3D animation, and Janet is an amazing example for this, and she has been an inspiration for me in so many ways. And there's also a whole field about data visualization. And the aim of this field is to show complex and maybe multidimensional data in an interactive and intuitive way. And this is super important, especially now that our data collection methods are getting more and more sophisticated. And people still have to be able to make sense of it to make sure we really understand what we are finding and seeing. So it was a huge inspiration for me to realize that this is something that's not only fascinating, but also incredibly important, both as a valuable tool for promoting my own research, but also as a legitimate career path on its own. And this brings us to the second point, which is how we use art to connect with the public on a deeper level and how we can inspire our youth to pursue careers in science. So ever since I saw my first microscopy image as I was studying, I was really blown away by the beauty of all of these intricate structures and all of these vibrant colors. And I thought it's such a pity that all of these really stunning images that we see under the microscope a lot every day, they very rarely get shared outside of the lab. So during my PhD, while I was waiting for the revisions of my paper to come back, I decided to try something. I went and I just booked my favorite microscope in the lab, but this time not for an experiment, but just for the sake of capturing the beauty of these cells that I was working with. So I picked some cool antibodies that stain all kinds of different cellular structures and create these patterns. And I thought they were so pretty, why not try to print it on maybe on a scarf or a dress and see what happens? And so I started wearing these clothes, and I was amazed about how often it sparked random conversations about science. So people were commenting on the patterns, on the colors, and I could tell them all about how these are cells that we are made of and what we scientists do and why science is important. And I was really amazed by how many people became curious about this and how I can use these visuals to connect with people on a different level. People who might be, intimidated by complicated graphs or figures, but they get excited about biology when it's presented in a more visual way. And this inspired me to create a whole fashion line. And I made clothes printed with microscopy images and with my own drawings. And it's been really fun to present this not only at conferences, but also in fashion shows and museums and outreach events, and seeing how this can spark fascination for a lot of people, especially students who may have never been engaged by science before. Yeah, but I realized that science communication is, of course, not limited to just drawings and fashion. And nowadays, we have so many new types of media that can take this to the next level. Especially, we have new technology that has a great potential to engage new audiences and just completely change the way we teach and learn science. So for me personally, the real realization came when I became part of this ifthen initiative. So the ifthen initiative aims to inspire girls to pursue careers in science. And the meaning is that if she can see it, then she can be it. And it basically aims to highlight scientists from different fields across all career stages. And they created this amazing exhibit that features life-size statues of all of these female scientists. So here's a picture of me next to my statue, and it's been an amazing way to interact with all of these kids and tell them about my research. And the aim is for girls to see that scientists can look just like them, and that there's no reason that they wouldn't be able to pursue a career in science. So through this awesome initiative, I was really excited when we learned that we would receive a small outreach grant, so for any project of our choice. And so I wanted to try something new and see if I can take my science communication to the next level. And I decided to create a video game. So I grew up playing games. I always loved how they kind of create these fantastical worlds that you can really immerse yourself in. But when I was in school, the idea to use games and interactive media in education was not a concept at all. And only after studying science, I realized that, so much of the magic behind all the things that make life possible often completely gets lost when you look at the textbook. You have all of these isolated diagrams, you have these complicated names to memorize, and all of this can seem quite intimidating, especially if you're a student getting into science for the very first time. So with this game, I thought I wanted to take the opportunity to depict the molecular world in a more visual and a more dynamic way, something that brings people closer to the amazing things that we see in our research every day. So I teamed up with a few people and made a game. So it's called Microscopia, and you can explore the inside of the cell. And what you can see here is the introduction sequence of the game. And you can see our character is wearing a lab coat and she's floating inside a vesicle through the inside of the cell and getting a first glimpse of this world. So you can see there was a mitochondria at the beginning, there's the nucleus, there are microtubules. And the difference is that everything feels more dynamic and alive than in a textbook. And we really wanted to create this immersive atmosphere by using these visuals, using engaging music to really bring this world to life. And the aim of the game is to allow the player to learn about the cell as they explore it, and also solve puzzles that are based on real molecular mechanisms. And some of these molecular mechanisms can be quite difficult to explain in a traditional way. For example, if you think about the mitochondria, you have this electron transport chain, and this is the series of reactions that create cellular energy in the form of ATP. And to do this, you have this whole complicated series of reactions that can be quite difficult to portray and quite difficult to teach in an intuitive way. So with the game, we wanted to give people the chance to experience this in a more engaging way without it feeling too intimidating. So I started with some sketching, and we wanted to take each of these complexes here and make a puzzle out of it so that you can learn about it as you solve it. And this is the final result. So this is the mitochondria level. You can, it has this basic kind of shape of mitochondria that people recognize. You can walk around inside with your character. And everything looks kind of industrial, like all of these machines, as a reference to the powerhouse of the cell, because this is what a lot of students are familiar with. And each of these structures matches the real shape of the protein, and the puzzles illustrate each of these chemical reactions. So here you can see two of these puzzles in action. So on the left side, you can see complex 4, and it converts oxygen into water. And you can see how these little yellow electrons here are bringing energy on the chain into the complex. And it's shown as a literal chain as a reference to the name electron transport chain. And then the player is adding these red protons to the molecule and turning the oxygen into water. And at the end, you can see little droplets of water dripping out of the faucet to illustrate that this molecule that they created is actually water. And as this happens, the protons from the matrix are getting pumped into the intermembrane space. And this is needed to power the ATP synthase. So the ATP synthase is here, and it's the actual machinery that generates the ATP. And we depicted it as it basically taking an ADP with 2 phosphates and a separate phosphate and pressing them together to generate the ATP, which has 3 phosphates. So we used a lot of these visual metaphors that combine these abstract concepts in science with things we know from everyday life. So players can really learn passively and without any prior knowledge of the cell. And this is an approach that we took for each of these puzzles. So here's another quick example. This is a puzzle showing the concept of dynamic instability in microtubules. So they constantly go through these cycles of growing and shrinking. And by adding these tubulin subunits, you can shift the equilibrium to make your microtubule grow longer and create a bridge to walk across. And after solving the puzzle, you receive a little trophy. And it has more information about the background. So players can do a deep dive whenever they are curious about learning more about it, but it doesn't interrupt the flow of the game. So yeah, that was the idea of the game. And we worked on this for like 2 years while also doing my postdoc. And at the end, we finally got to share it with the public. And we got to present it during the San Diego Comic-Con at the Comic-Con Museum. And it was a really, really amazing opportunity to watch people play it for the first time and also see how students interact with it outside of a classroom setting. And it was amazing to see that kids were actually choosing to play the game. So there were so many other cool exhibits, but people were still coming and playing the game. And some kids were there, they wanted to keep on playing, even though the parents wanted to leave. And it really demonstrated that we can make science interesting and exciting by using this interactive media and new technologies and get a whole new audience excited about science. Yeah, and oh yeah, by the way, for those who, thank you for the comments in the chat. For those who want to try it, you can play it on Android, iOS on the browser, and it's free to play. It's pretty short. So if you want to try it out, please go ahead. And I'd love to hear what you think. Yeah, but what I wanted to say is that through kind of doing a lot more science outreach is, I realized that despite all of the conferences I've been to and all of the amazing scientists that I've had the pleasure to work with, some of the really most impactful interactions I've had is actually sharing science with young scientists or with young students. So every time, I participated in an outreach event, there's been at least one of those aha moments where you can see that you sparked something like a passion or an interest or a curiosity in science. And, it's easy to forget because many of us were lucky to grow up in places where we always knew that this was a career option. But there's also a lot of young people who don't have that. And this is why it's really important to remember this side of science communication as well. And, you're speaking to people outside of science, you're sharing your passion, you're really sharing your knowledge with them. And there's a chance that you might be the person who changes somebody's life because you inspire them to become a scientist. So these experiences have really been rewarding. And I definitely recommend giving this a try. Even if your main passion is doing research in the lab, it really changes your perspective on science and what you're doing in the lab every day. It really gives it a much bigger perspective. Okay, so that was the part about how I discovered science and art and how I kind of started experimenting with it. And now I'd love to tell you a little bit more about the process behind how I come up with ideas for my drawings and then how I also create the illustrations. One of the questions that I get asked a lot is, how do you translate a text from a manuscript or a paper into an artwork? And, we all know that very often concepts in biology are abstract and hard to imagine. And my approach has been to just take these really complex elements and put them into a familiar context that anybody can relate to. So just like we saw with the Lego pieces, like we saw with the mitochondria level inside the game. But of course, some things are easier to illustrate than others. And there are a few tricks that I use to help me get ideas for the concept. So here are some examples. So for example, you can incorporate real scientific data and you can use protein structures, microscopy images, or all kinds of data. You can also try to highlight some interesting facts about some aspect of the story that makes it more memorable and easier to connect with. You can also just take a model figure from a paper or graphical abstract and create an artistic rendering of that to go along with it. Or you can also find, some sort of analogy or metaphor for a complex molecular mechanism that then translates to something that people can better relate to. And you can also just take a play on words or a gene name and use that as a concept to get an idea for an illustration or an artwork. So I'd love to show you a few examples to illustrate each of these points so that hopefully it will spark some inspiration for you as well. So, for example, you can use real scientific data. So for me, the starting point is often some sort of scientific paper, like for example, this paper on mitochondrial ribosomes. And here, the authors wanted to understand how the structure of these mitoribosomes has evolved over time. And what they did is they compared the protein structures in different organisms and found that over time, there have been structural changes that create little instabilities inside the ribosome. And to compensate for this, the cell patches these instabilities by incorporating tRNAs into the structure. So just incorporating these tRNAs to make sure that the ribosome stays stable as the structure is evolving over time. And so I started thinking about what kind of analogy could I draw to make it easier to illustrate. And I had the idea to make a reference to crocheting, because to me, the RNA kind of looks like a string that is woven into the structure, and it kind of reminded me of that. And I also love crocheting, so I was a little bit biased. And here's the final result. So what you see is you have two hands crochet this structure, and the structure is blending into the ribosome. And to keep things scientific, I chose to use the real protein structure. And I also make sure to highlight the classical fold of the tRNA, just like we know from textbooks and representations. And the structures are labeled by the color of the yarn. So red is the RNA, and the other colors are the protein elements of the structure. So basically, it's real scientific data fused with a metaphor to get the point across in a more intuitive way. And you can also use other types of data. For example, Hi-C data. This is a method that measures chromatin interactions in the nucleus in 3D. And these plots that usually people make of Hi-C data always somehow reminded me of a landscape with pyramids that are kind of reflected by water. And so in this illustration, that's the reference that I used. And they found in this paper that gene expression is not affected even when you have rearrangements in the chromosomes. So in this drawing, the pyramids represent the normal chromosomes on the top, and the reflection in the water shows the rearranged chromosomes. So you can see that the reflection is not symmetric. But the landscape overall is not changed, kind of symbolizing that the gene expression is not different. And if you look closely, you can see parts of the actual data overlaid with the drawings of the pyramids. So yeah, this was another example to show how you can use real scientific data in your illustrations. And of course, this is not always as easy. It depends really on the project and on the paper. So a different approach could be to use some sort of interesting reference that connects with the data, with something that people are familiar with, something that catches people's attention. And so, for example, this paper describes a selection method for CRISPR that is based on introducing a mutation, and it makes the cells resistant to a certain drug. And the way it works is that you have your CRISPR-Cas9 system, and alongside introducing the desired mutation in your gene of interest, you also mutate the ion pumps in the cell, and you select them with a drug that kills all of the cells that don't have the mutation. And so only cells that have been edited by CRISPR will survive, and the others will die. And it turns out that the same molecule that was used, that is used for this method, was used as an arrow poison in the past. So I thought I'd make a reference to that. And so I made this drawing. You have the cells and you have the arrows. And the arrows represent this molecule. And you can see the chemical structure indicated in the feathers of the arrows. And you can see that they are killing the red cells. So these are the cells that don't have CRISPR in them. And they are piercing through the ion channels to kind of kill the cells. And the blue cells have the CRISPR-Cas9 inside the nucleus, and they are resistant to the drug, so they are protected from the arrows. Yeah, and another way to get some idea for a concept is to use a model figure or a graphical abstract from a paper as a starting point. So this is a paper from my previous lab where I did my PhD, and it's about cross-linking the chromosomes after anaphase so that the nuclear envelope can form a single nucleus around it afterwards so that they are not scattered. And this is mediated by a protein called BUF. And what it does, it cross-links the DNA strands on the surface of the chromosomes and then creates this network so the nuclear envelope can't go in between them. And so I used the color scheme from the model figure to show the DNA here. And the DNA is being knitted into this network, and it's held together by these blue beads, which are representing the BUF protein. Okay, and two more. So this one is an example for a paper that describes a pretty complex mechanism. And it goes very much into detail, and it can be quite tricky to kind of come up with how to draw this. And in this paper, basically they describe how mitochondrial DNA switches between replication and transcription. So when you have the RNA polymerase transcribing the RNA, when it reaches a certain sequence, the RNA forms a secondary structure that stops the polymerase from going any further. And this makes room for replication. But when the polymerase is bound to this factor called TEFM, then it can overcome this barricade and continue transcription. So the presence of this TEFM determines whether there is replication or transcription. So the metaphor here is you have a train. It's going along the circular DNA of mitochondria. And you can see that the RNA that comes out of the train forms this barricade. It's basically symbolizing the secondary structure of it. But the train has this cowcatcher here in the front, and it allows it to break through the barricade. And this allows the train to continue transcription. So yeah, basically you have the same information as in the graphical abstract, but using metaphors that are more intuitive to connect with if you're not familiar with this field. And finally, you can also just use a simple play on words to describe a paper. This one, for example, is about a computational method that helps to find very rare mutations in human populations. So it's like finding a needle in a haystack, but the haystack is a chromosome and the needle is the rare mutation that is being found. Yeah, so those were some examples. And overall, I would say for me, the trick has been to identify something that catches the people's attention and then add elements to make the artwork interesting on multiple levels. So scientists, they might see all of the detailed references, and non-scientists might still appreciate the general concept without feeling intimidated by too many details and complicated graphs and things like that. And I found it, as I was experimenting, I found it really powerful to even to communicate between scientists from different fields, to complement these traditional scientific figures, and also share these concepts in the forms of outreach and education. Yeah, and another question that I get a lot is, how do you actually create these illustrations? Are they hand drawings or are they digital? And how do you add the colors? So I want to give you a very brief overview of the technique that I use. And it's of course, absolutely not the only way to do it, and probably not even the best way, but it really works for me, and I hope it can give you some ideas as well. So here's an example. I used a microscopy image of a cell going through anaphase as a starting point, and I created this drawing showing how the chromosomes are being pulled apart by the mitotic spindle, which is represented here as ropes. So the first step for me is creating a sketch. Sometimes it's one, sometimes it's many different sketches, but it really helps me experiment with the layout and the composition of the artwork. And actually, most of the time that I spend in kind of the most challenging and most exciting part is coming up with a concept and making these first sketches. But once I have a sketch that I'm happy with, I create a detailed pencil drawing. And so I create a pretty big drawing with real pencil on paper. And I really try to focus on using shading to create kind of the depth and to make it look as realistic as possible and kind of as detailed as possible. This is one of my favorite parts of the process in creating illustrations. And then I go to one of these printing places where they have these big scanners, and I scan the drawing and import it into Photoshop. And now I can add the colors digitally and overlay it with kind of the shading of the drawing. And the first thing I do is I first start by removing the white background. And for this, I create a mask around it. So I don't want to go too much into the technical details. There's probably many ways to do this. But basically, what I do is I kind of trace the outline of the drawing on a digital tablet. And in this mask, everything that's black is going to be transparent, and everything that's white will still be visible. So adding this mask to the drawing with a black background behind it ends up looking like this. And now the next step is adding the colors. So I use a similar technique for this as well. So I draw a mask around just the chromosome part and add a color to it. And the blending is set in a way that everything that was white in the pencil drawing will show the underlying color, which is blue. But everything that was dark still stays dark. And this is how I kind of blend the shading of the pencil drawing with the colors that I add digitally. And I can do this with each part of the drawing and add colors to the hands and to the ropes and whatever other elements are there. And now comes the fun part. Now I can start playing with adding more colors and add some shading and some highlights to each of these parts. And this is what the end result looks like. So you have the pencil drawing. It's still fully visible, but now it's overlaid with all of these colors and shadings and highlights. And if I were to remove the drawing from this is what it would look like. So this is just the colors and the shading and everything. And this is what it looks like together overlaid with the drawing. So that's basically the whole process. And the great thing about this is that the way it is set up, I still have the flexibility to change a lot of different things. So I can experiment with different backgrounds, with different color schemes. And I generally like creating multiple versions to see which one looks best. I can never decide which color it's going to be at the beginning. So this is a fun stage to play with it. And this is also where I add real scientific data or textures and just play with it until I'm happy with it. So yeah, that was the workflow. I hope it was helpful. And again, it's by no means the only way to do it. Some people love drawing fully digitally. Some people love painting. And the best thing about art is that everybody can have their own technique and just bring their own style into this. Okay. And in this last part, I would love to share some advice on what you can do if you want to explore the world of science and art. And this is really some advice that I wish I would have given to myself at the beginning. I really felt every time I felt like I had no idea what I was doing. So I hope it will be helpful to some of you as well. So the first thing you can do is try to find a creative way to explain your own research. And you can really experiment with any medium that you're passionate about. It doesn't have to be drawings or fashion. It can be anything. And try incorporating it into your science. For example, you can add a drawing to the title slide of your presentation. And you can use it to introduce the context around your research topic. Or you can use it to explain your new model that you came up with based on your data. Or you can even use it as a graphical abstract or support a grant proposal or maybe an application for an award, and see how this helps communicate your science and how it can engage different audiences. One thing that I also really like doing is presenting my project to non-scientists and really get feedback from them. And this has been super valuable because sometimes you just need an outside perspective, and it's easier to connect with other scientists that may be less familiar with the background behind my research topic and things like that. So there's so many different fields in science, and even communicating between these fields is really important and sometimes really challenging. If you are a PhD student or a postdoc, and you're curious about experimenting with it, maybe try collaborating with your lab or your institute on creative projects. So this is what helped me a lot when I was starting out, because especially when I was transitioning from primarily working on the bench to doing both. And this was a great way for me to know that I'm bringing value to my lab, not just by doing experiments, but also by helping promote the work that is being done in the lab to the outside world and to other labs. And so, yeah, for example, my first official art project was creating a banner for my lab's website. And then my first journal cover came from collaborating with a lab member when they were asked to submit an artwork. So definitely ask around. Maybe your lab needs a logo like my current lab did. Or maybe reach out to your communications department in your institute and see if there's anything you can collaborate on. For example, by making an illustration for an event or press release or conference. And all of these little projects really helped me at the beginning. In retrospect, I realized that it was a really great way to work on some projects where you have some level of accountability to make sure it really happens, but also it was a very low pressure environment. So it allowed me to really gain more experience and establish kind of a workflow for creating illustrations. Also, experimenting with different techniques and also learning what it's like to work with different types of people. And all of this has really allowed me to gain more confidence as I was exploring this field. And networking can also be extremely helpful to connect with people and learn about creative careers in science. One of the most amazing experiences for me was when I discovered that there are conferences that specifically focus on science visualization. And the VISB conference is actually a great example. So when I attended it for the first time, it turned out to be an amazing way to connect with others in the field. And I met people from all kinds of different backgrounds. So there were scientists who started exploring visualization, or who became illustrators, or who went into animation. There were artists who started getting into science, and lots of interesting careers that I had no idea even existed. So I really recommend going to one of these conferences, even if you're not making illustrations yourself. It's also a great place to learn about how to present different types of data, and you can discover new visualization tools that your research can benefit from. And it's been a really amazing learning experience for me too. And I would say the most important advice I can share is to volunteer for things. So at the beginning, I felt like there weren't many opportunities, but as I started looking for them, I realized that they are there if you ask around. And each opportunity opened several new doors, and everything just has grown exponentially from there. And so here's an example. For example, a colleague of mine was organizing a symposium in Mexico, and they were looking for speakers. So he asked me if I wanted to present my science there. And I said, of course, but maybe let's also try to do a science art show and see what happens. And so we decided to ask some other scientists and artists to exhibit some of the artwork, but we also wanted attendees to be part of it. And so we decided to leave a blank wall and asked people on the first day to bring their art. And the next day, no one could believe the amazing artwork that had filled the wall. And, people were bringing their drawings, pictures, and even sculptures. And they made all of this at home and never even intended to show to anyone. And it was just fascinating to see that all it took was an open space, and people were sharing the creativity, they were being appreciated for the talents, and realizing that they were really surrounded by like-minded and creative people. So I found that incredibly inspiring to have these types of people in our community. And yeah, so that's it from my side. As a take-home message, basically, I want to encourage you to follow your passions and see how you can combine them with your research. And it will really change the way you communicate your science, and perhaps even open up a new and completely unexpected career path in the future. And yeah, I have so many incredible people to thank for supporting me as I was starting out, for giving me the freedom to continue pursuing this passion, for being such an inspiration, and for some amazing collaborations that came from that. And yeah, before we go to the questions, as Janet already kind of mentioned, I have one quick announcement to make for those who plan to attend the Cell Bio meeting in Washington in December. And so we'll be organizing a subgroup. It's called Science and Art, Bridging Two Creative Universes. It will be on Saturday, and we have 13 amazing speakers, including some people who have been an inspiration for me for a really long time. So I'm super excited for it. And we basically want to discuss the impact of artistic practices on scientific research, on science communication, and also on education. And we just want to start building a community of cell biologists who use art in their research and in their communication. So if you're planning to attend, I hope to connect and see you in person. And yeah, that's it from my side. Thank you so much for your attention. Thank you so much for coming. Thank you so much for all of your lovely comments. And it's been a pleasure, and I'm really happy to take questions. Thank you all.
You should definitely check out the chat, since there are tons of kudos there and a lot of people really appreciative of this beautiful talk you just gave. So thank you so much. Yeah, so let's get into some of the questions. So the questions, I think, we'll start with the ones that are a bit more specific about some of the things you talked about. So there are a couple game related questions. Can you tell us again how to get access to it, to the game?
Yes, absolutely. So the game, you can just, I'm going to put a link in the chat. It's called Microscopia. And all you need to do is just go to the website and there are links. Or you can, if you want to play it on your phone or your tablet, I think it looks best on tablets. You can go to the App Store on Android or iOS, just type in the name and you should be able to find it. And it's free.
Yes.
You can play it. It's not super long. It takes about maybe 30 minutes or 40 minutes to play it. So yeah, if you do, I'd love to hear what you think. I'm about to put the link in the chat so you can have it. But yeah, I hope you all enjoy it.
Yeah, and I just Googled it and found the link really quickly. So that's another way other folks can find that. There's a question about what are your favorite comments from people who played the game, and did anyone say anything that surprised you? And, you know, kudos again on the game.
I love that question. Yeah, so it's been a really interesting experience because as we were making the game, we did a lot of surveys. We kind of collaborated with teachers and classrooms to see what people were saying. And we heard some amazing things. And I don't have anything specific in mind right now. I guess most of the comments were just saying, you know, how they really loved the colors and how engaging everything was, how different it was from textbooks. And yeah, most of them have been really positive, and we also learned a lot. So from these comments, we also learned that, you know, adding these trophies, making sure not to interrupt the flow of the game. And then also things like adding, there's this, we want to make sure it's not frustrating, but we wanted to make sure the puzzles are challenging. So we added like an option where you can have help. So it gives you a hint how to solve it for those who want it, to make sure that the experience is not frustrating, but still challenging. So yeah, a lot of comments based on that. And yeah, we learned a whole lot about making games and things like that. And yeah, overall, a really positive and interesting experience.
Great. There are a few questions that are kind of touching on kind of the technical aspects of, you know, what kind of the software, the specific software you used. I know you mentioned Photoshop. Is there anything else?
So for me personally, I am super familiar with Photoshop. So that's basically what I've been using. I know there's so many other amazing software that can do basically the same thing. And for me, I think the reason why I use Photoshop is because I've been familiar with it. And I have this Surface computer that comes with a pen. And it's basically like drawing on a tablet. But I'm sure you can use, you know, Android-based apps or iOS-based apps for this as well. I think it really is just, for me, historically, this is what I'm most familiar with. But I think the concept of, you know, having these masks and having the blending mode set to a way where you can kind of blend different colors and different shadings is probably really universal to a lot of different apps too. But if anyone has a specific question, please reach out and I'm happy to go over it and show you exactly how I do it.
Great. So there are a few other questions, kind of more general, about like, and this is, I think, something I would love to hear too, is about work-life balance and how do you manage to, you know, you talk about doing everything simultaneously, but in practice, was that easy to do when you're training or, you know, yeah. So a work-life balance question.
Great question. I was afraid this question was coming, but I'm also excited to talk about it because for me, it's been kind of, especially at the beginning, a little bit of a struggle because the fact is, you know, I got so excited about all of this and I'm also excited about the science. So in the end, I just ended up doing as much as I can. But I know, so like over time, you know, I talk to a lot of different people and, you know, there are people who do just art, who have transitioned to do art. And then there's also people who do both. And, you know, everyone has kind of a very unique situation. For me, I would say it's been a little bit easier now that I've learned to say no to things and things that we can all probably learn that over time. But I would say that it's possible. It's not super easy to find that time, but I do it because I love it. And I get to try so many things. And also I see it as a learning experience. So for example, when I decided to work on the game, I knew it would take a ridiculous amount of time, but I learned, you know, how to draw on a tablet. I learned how to, what it's like to come up with such a big project and make sure it gets completed, deal with the budget, put together a team and things like that. And I feel like it was really worth it in retrospect. And because the way I see it is now is the time when I'm exploring and I know I won't be doing it in this form indefinitely. So it's a great way for me to kind of get a little taste of all of the different things and make career decisions based on that in the future. So I think for me, right now, the science and art and outreach has been all kind of separate, but I'd love to be able in the future to find something that where I can combine it all into my main work. So I think no matter what we end up doing, I can't see myself not doing science or art. But yeah, in terms of balance, I would say I think one of the things was to really learn to really pick the projects depending on what I want to learn, what I want to take out of it, what I'm most excited about. And yeah, and then really try to be efficient with time management and also make sure to take breaks sometimes. That's also something that I've learned is to, there's always something to do. There's always going to be exciting project, but the truth is your health is also a priority. So kind of going between waves of, high intensity and the lower intensity really has helped me to kind of get a little bit of a balance.
This is a great segue for a couple of questions that are dealing with next steps. So what are you thinking for your next steps for your career? I guess a next project, kind of immediate and long-term kind of future.
Yeah, great question. Yeah, I'm also wondering that myself. At the moment, I don't have any specific plans. Right now, I'm focusing on my next publication. So I'm very excited about that, to get that published. And it will definitely take priority right now and time after, all of this time working on the game. And, I stopped doing commissions for a while just to make sure that I'll be able to focus on one thing. But yeah, it's a great question because I feel like I've been balancing a lot of really cool and fun things. And I hope that I'd be able to find something, like I said before, something that would allow me to combine all of this into one thing. And I feel like a lot of people I talk to, actually including you, Janet, I feel like a lot of people who are in really interesting positions, it's not like you go and apply for something. It's basically you make your own position based on kind of the expertise that you collect over time. So that has been a really great inspiration for me. And I'm trying not to kind of narrow down like exactly what I want to be doing, but more keep an open mind and talk to different people and see what kind of options there are out there. And again, going to the WISPE conference has been really amazing because there are a lot of people with unconventional career paths and unconventional kind of steps that they took. And many people that I talked to, a lot of the things came through serendipity and kind of coincidence. And it's not something that you can really plan exactly in advance. So I just want to make sure to keep my open mind and see where it leads me. Sorry, yeah. I don't have anything more specific.
No, I think that's perfect. This also kind of relates as well. There's a couple questions about a community, art and science communities, and platforms where you share your art with scientists and non-scientists as well. You mentioned WISPE. Are there other kind of communities or platforms that you use?
Yes, so there's a lot of actually communities that I feel like almost every day I connect with a person and realize that I feel like there's a lot of people having these really awesome small communities kind of in their kind of university or in their circle. And specifically, I guess there are so many, I don't even know which ones to name, but I think the WISPE is one of them. I think that's one of the big ones, especially in terms of more data visualization related. But then there's also communities that focus more on science outreach and using art for education and classrooms and outreach. And then there's also communities that combine science and art. So there are these, often exhibitions and things like that. Basically, what I noticed with these communities is they're all awesome, and all of these people organizing them are really inspiring. But I feel like there's not really a bigger framework, but something that would kind of be able to get all of these people together and create something that can, be scaled and really make sure that it's not so isolated. So that's one of the things that we want to do with the subgroup, is to kind of start building a community, start kind of coming up with ideas how we can make it easier for scientists to collaborate with artists, or scientists to make their own art and kind of share it on a bigger scale. Yeah,
I think taking advantage of the society is a great way of building a community. I guess in terms of like getting your artwork out to the kind of more general audiences, are there any specific platforms you use for that?
Yes. So for me, one thing that really helped me with my outreach and all is, one of them is the ifthen initiative. And if you reach out to any of these women that are represented there, they're all engaged in some sort of outreach in some sort of form. So having kind of this network, and we are all, super open to collaborations. And so if you feel like you want to get started with that, please reach out to one of us. The other thing is, I started volunteering with this organization called Young Women in Bio, which is a national organization. They have different chapters in different places. And so what we do is basically have a group of volunteers from biology, biotechnology, and other fields. And we love creating these outreach events. So like collaborating with an organization or going to some of these STEM fairs and just having, a table with activities. I love to bring my little microscope and some drawings or the game. And that's how I've been basically able to do most of the interactions with, kids and people visiting, people who are excited about science or people who want to learn about it. And so that has been something that I never really expected to be doing in the past, but turned out to be really inspiring and really gave me a new motivation to continue doing my research in the lab. So basically volunteer with organizations and reach out to other people who inspire you, people who are in this network. There's a lot of people who, know teachers or educators, and they're all looking for new ways to engage their students. It's been just, collaborating with different places and trying out different things.
Great. And in terms of you and like where you share your work, do you, I guess there's a question about where to follow you. Are you on Twitter? I know you're on Twitter, but are you also on some other platforms like Instagram or any of those other,
Yes, absolutely. So Twitter is what started everything for me. So I made a Twitter account and started posting my online. And this is how I kind of started connecting with the first people, with people who randomly needed an journal cover and things like that. But I'm also on other platforms. Let me actually put on the previous slide. I hope you can still see them. But on the bottom here, you can see my contact and also my social media handle. And so I'm on Instagram, I'm on LinkedIn and on Facebook. And it's already been really overwhelming to deal with all of those platforms. But I also think it's really important because that's how you can connect with other people. There are students reaching out to me who have been curious about this, but never really were able to take the step. And just a really great way to really communicate with these people and really connect and help them, and also get inspiration from other people, connect with organizations. So definitely Beata Science Art on all of the platforms.
All right, maybe we have time for one more question. We have a question about, do you have advice for scientists who want to commission artwork on how to get funding to do that?
Yes, great question. So for me, I'm usually, coming from the other perspective where I collaborate with labs, and it seems like often they have funding. And I think from what I was been talking with a lot of scientists is that, a lot of the grants that you can apply for science funding also have an outreach component. So often increasingly you kind of have to say, like, how will you communicate your science to the outside world, and how can you make an impact in your community? And so I think some of them have been able to use this sort of budget for outreach projects and things like that. I think, organizations and people seem to be more and more open to it. I also feel like institutes, when they, do the communications, seem to have some funding for that as well. So sometimes, I create drawings for press releases and things like that. So this is from my side, what I've been seeing, kind of the transition from, drawing for my friends and then working for other people has been a little difficult for me because I think it's a whole other mindset, want to get outside of basic science. But I feel like even if it's just a little thing that you ask, it's just makes sure that what you're doing is valued, even if it's just symbolic. I feel like it really helps. But it seems like increasingly, I feel like people have been able to get funding for it in some way.
Yeah. The National Science Foundation, the outreach component, I think, is a great way. I agree, to get funding for doing this type of work. I think that might be all we have time for today. There are a couple more questions that hopefully you can take a look at, Beata, after we close out here. But thank you, everyone, for joining us. And yes, please do consider coming to the talk. I guess it's the user group symposium that Beata mentioned, as well as our roundtable discussion to talk with other people who are interested in visualization in biology. Thanks, everybody.
Thank you so much again. It's been a pleasure. And please reach out to me, and I'm happy to chat more about anything in the future. So my email is down there on the slides. So yeah, please reach out, and thank you again for everything.
Thank you so much, Beata. This was so inspirational. All right. Bye, everybody.